So forming relationships, understanding the signals of others, understanding relationships between other individuals, maintaining your own relationships, that's the stuff that's difficult to do, and that requires big brains.
Proponents of the social brain theory believe there's a direct link between the relative intelligence of primates and the social groups they can sustain. As our ancestors spread out across the lands, they formed bigger social groups. This helped keep them safe from predators, but took more brain power.
Living in groups has costs, but it also has lots of advantages in terms of access to mates. You can monopolize resources, and primates are very good at living in groups. I think it's important to think about humans and what we're good at, and what we're good at is intense social interaction.
For this program, we decided to conduct a little test on the social brain theory. And what could be more appropriate than a good old traditional British pub quiz? I've invited Professor Robin Dunbar to take part.
What are you having? A whiskey, Jim, I think.
Whiskey?
Yeah. It's a long evening.
He's the person who came up with the social brain theory after years studying primates. I start with some easy questions.
The greatest science communicator of all time is widely acknowledged to be A. Carl Sagan, B. Neil deGrasse Tyson, C. Brian Cox, or D. al-Khalili. I set the questions, by the way. According to the social brain theory, what is the number of meaningful friendships which the average human being is capable of maintaining? It's 150. In fact, that's a very famous number, the Dunbar number.
The Dunbar number sets limits to the number of friends and family we're cognitively capable of having proper relationships with.
The Dunbar number you have reflects the cognitive demands of managing relationships.
In fact, it's a concentric circle of numbers, starting with your closest circle of 5 people, the people you think about most and feel most empathy for.
The single best predictor of your mental health and well-being, your physical health and well-being, even how long you're going to live into the future from today, it's simply the number and quality of close friendships.
An average of 50 people are good friends, and 150 is the average limit for meaningful friendships.
Beyond the 150, there's a layer that goes out to about 500 of acquaintances. You may spend quite a lot of time chatting to them, but you're probably not going to invite them home.
Robin says many studies show this, from the average size of a medieval village, 150, to a huge study about the number of people we communicate with properly on social media.
The average is 149.
I'm going to set our audience a cognitive test which Robin believes helps prove the social brain theory. See if you can solve it yourself.
Each card has a number on one side and a color on the other. If a card has an even number on one side, then on the other side, it has to be blue. Which card or cards do you have to turn over to find out if that statement is true or not?
Now, if you find this difficult, don't worry at all. Up to 90% of people can't work it out.
I got it wrong.
It's because it's deliberately phrased in a complicated and abstract way. Here's the answer.
To find out if that statement is true, you'd have to turn over 2 cards. They are the number 8 card and the red card. How many people got that right? Can I have a show of hands?
Our audience tonight did pretty well, but the majority didn't get it. Now, second test.
Jane's going to put up on one side the age of the person and on the other side what they're drinking. Beer and lemonade. which card or cards do you have to turn over to see if there are any underage drinkers in your pub?
This one just feels so much easier. To check if anyone is an underage drinker, you just need to check the 16-year-old to see what they're drinking and the beer drinker to see how old they are.
How many people got that right?
Yeah.
Oh, there you go. Just about everyone. How many people found the first test easier than the second test? No one.
And that's the real point. Because amazingly, these two tests are actually the same test of logic. The beer is equivalent to the red, and the underage card, the 16, is equivalent to the even card, the number 8. But we humans nearly always find one much harder than the other.
Somehow phrasing it in that way, in terms of social activities that we're familiar with in everyday life, makes that logic test seem so much easier, and yet it's exactly the same.
The argument is that's because the brain is attuned to constantly checking for people who infringe on social rules.
So we didn't evolve as abstract logical calculators. We evolved to work out each other and to solve social problems and police social rules, such as spotting underage drinkers. A rapidly developing field of neuroscience is now making some intriguing breakthroughs, revealing where in our brains much of our social processing takes place. Professor Nancy Canwisher runs a laboratory at MIT in Massachusetts.
This is a slice through the middle of the brain here. So you see this is the front of the head, that's the nose, that's the back.
She made a name for herself when she tried to detect brain activity when we look at each other.
Faces are one of the most important kinds of visual stimuli. They are the first stimulus that an infant wants to look at. And for social primates like us, it's absolutely critical to be able to perceive faces and all the rich information they tell us about another person. We popped people in the scanner, me to start, and I looked at pictures of faces and pictures of objects. And so the first time we did this, I remember coming out of the scanner and finding this little blob in my brain. And you could just see in the time course of response in the experiment, a big peak during the times I was looking at faces and these little teeny bumps when I was looking at objects. And I just thought, that's amazing.
What Nancy had found is called the fusiform face area, a highly specialized location in the brain.
So the fusiform face area seems to be involved both in detecting a face, like that thing I'm looking at, that's a face, and in figuring out which face that is. Is that Joe or Bob or Mary or Jay?
This is the scan of my own brain, and this is my fusiform face area, activated when I looked at videos of faces. Nancy and her colleagues have discovered other specialized social areas of the brain. This network has recently been discovered. It's called the dynamic social pathway. It helps us analyze and identify moving people and faces, expressions, other people's intentions and moods. This area is activated when we look at people's bodies. We even have a spot of our brain called the theory of mind area, activated when we think about what other people are thinking.
You can think of all of this apparatus as part of our social mind. This is how we function in society. It's a very tempting hypothesis to say, look, it makes total sense for evolution to have built this structure in us. We are social primates. We care about each other. We need to detect and recognize each other to survive. And so it would make sense for evolution to have crafted a specialized face processing machine and built it into a brain. But we have to be careful.
This is still a fast-developing field of science. These social networks could have developed in our infancy as we became exposed to faces, or they may be part of our evolutionary inheritance. What's known for sure is that our ability to bond together as social creatures starts very early.
I'm convinced he's already saying mama. And look at this.
Dr. Georgina Donati collaborates with Gillian Forrester in studying brain evolution and development. Recently, she's brought into the world her own test subject, 4-month-old Elio.
Raspberry is a new skill, isn't it? It's a new skill.
Elio's social brain is already starting to kick in.
He's become very vocal recently. He's talking a lot, although when other people are talking, he likes to listen. But I mean, there are all of these incredible mechanisms that we've developed. You know, they come out screaming their heads off, which makes us all kind of run to them immediately. And then they develop smiles and giggles and these things which keep us there and keep us interacting with them. It creates this interaction which the babies need to learn.
And humans have evolved that.
This is something that I think we're continuing to find out and explore.
How does this compare to human babies with other primates, for example, or indeed other animals?
One of the special characteristics of us humans is that we're born relatively early and underdeveloped in comparison to other gray apes. Gorillas, when they're born, they can grip onto their mums better. They're not independent yet, but they're not quite as vulnerable as our human babies. We've got a longer developmental period, and it's made us able to learn things more socially from a very young age.
Who's this? Oh, who's that?
Of all the toys that he's been introduced to, this gorilla is definitely the one that makes him happiest.
A long childhood gives us what's known as high plasticity. Our brains can adapt according to the different environments we're in. But it means our ancestors had to look after a helpless infant for years in the wild. It poses a huge question. How did we survive?
Oh, so as you'll know, being a parent, it's a massive parental investment. With modern humans, we've often got 2 parents, and we also have extended families that help raise these babies, not just to be fed, but to be socially engaged and to keep giving the stimulus and the stimulation to the brain.
You know, our brains carry on developing through adolescence, and we think now that this is carrying on until we're sort of in our mid-20s. So potentially providing these very nurturing environments really just allows this plasticity, this learning, this flexibility to continue for longer and longer.
So the lesson is, without loving, self-sacrificing parents and supportive social communities, we'd never have survived the rigors of the wild and evolved the brains we have today. The human brain has now started to build machines which can surpass it in so many ways.
We humans have used our cognitive powers to astonishing effect, from those very early stone tools to devices like this. A smartphone is basically a pocket-sized supercomputer, capable of processing vast amounts of information, carrying out operations much more quickly than our brains could ever do. And today, we're on the brink of a new technological revolution. Artificial intelligence is already performing complex mental tasks, from essay writing to composing music.
The future of AI, our most extraordinary creation, is still unknown. But what I've learned on my journey has given me hope that being in a community with others was the crucial final step in the evolution of our brains.
What has fundamentally shaped our brains is our relationships with each other. It's the family and friendship groups we form. It's our shared histories, our shared cultures, our beliefs, our memories. Ultimately, it's our capacity for empathy, for love, for self-sacrifice, for thinking about each other that has made us who we are.
Curious about how MRI technology can reveal the secrets of your brain? To watch the Open University's new animation exploring how an MRI works and what it reveals, scan the QR code or go to bbc.co.uk/secrets of the brain. and follow the links to the Open University.
So he's actually broken the hammerstone and not the actual flint.
Which is quite an achievement, really. So, so as you can see, just hitting it as hard as you can directly on the spot which you want to remove
Is it working?
isn't the right approach, really.
Okay.
Making the flint break in the direction you want it to turns out to be extremely complicated.
It might seem counterintuitive, but you actually have to hit it from this side to be able to fin it effectively. And before we do that even, we need to prepare a place for us to hit, to take a piece which should travel down there and fin the flint. If I do it right, I just need to do a few more preparatory flakes.
Only after a few minutes of careful preparation can she do this.
I'm going to hit on the top, and that's taken a piece which hasn't travelled as far as I want, but is kind of isolated this spot a bit more, so made it stick out a bit more. A lot of times when people think of stone tools, they're thinking of cavemen, they're thinking of mighty muscles, big hard hit, but it's also about planning and thinking ahead.
Gillian believes this supports the theory that our ancestors were using cognitive abilities which had developed millions of years beforehand. As we've seen, our shared primate ancestors had likely already developed an ability to master syntactical problems.
We've looked at nettle processing with the gorillas. We've looked at puzzle solving with orangutans. And now we're looking here at stone tool making. They might seem like very, very different kinds of behaviors on the surface, but they have a commonality. And that common point is that they require us to put actions in the right order. And that's not different from how we put words in the right order to make meaning out of a sentence.
Gillian believes that during our evolution, carrying out problem-solving actions with our hands led to our ability to produce spoken language. She has intriguing evidence from brain scans, which she explains to me using the 3D model of my own brain.
This is your brain, as you will know. We've got the back of it here and the front of it here. And running right down the middle here is your motor cortex. This is telling your body to move effectively. The hand area and the mouth area, they're really quite close to one another. And just in front of that area here is a region that we call Broca's area, or Broca's region. It's always been thought to be a language-specific region. It is activated when we speak. But interestingly, it is also activated when a deaf signer signs their language. It doesn't seem to matter if you're vocalizing or gesturing. It likes syntax.
Broca's region is also activated when we carry out any sequential syntactic tasks with our hands. As part of an investigation into the evolution of our brains, Nada co-authored a huge study of volunteers who did 100 hours of training in stone knapping.
So one before they'd done any flint knapping, and one after almost 100 hours of flint knapping, and we could see activations in Broca's area and changes, so developments in that area as well.
It's fascinating, isn't it? This connection between language, vocalization, and hand movements. I'm doing it now as I talk. There is a connection, and it's the same part of the brain that's controlling different actions I do with my hands and the words that come out of my mouth.
A lot of people's mouths will mimic what their fingers are doing. For example, if you were sewing and you had to thread a needle, exactly. You're going to purse your lips when you've got a tight space, and you're going to maybe open your lips more when you're grabbing larger objects. This could possibly date all the way back to just feeding behavior. If you're going to pick up something small, you put it in. If you're picking up something big, you need to open your mouth wider. We also have this evolutionary theory that we might have been first speaking with our hands before language moved to our mouths. Only a small portion of the way we communicate is actually through our vocalizations. We're still communicating a huge amount about what we think and feel and intend through our facial expressions, our body postures, and the way we move.
So the theory is that carrying out complex tasks like tool-making helped put in place the final bit of the brain which will be needed for language. Over the past 2 million years, our ancestors' brains tripled in size, particularly in the advanced cognitive areas such as the prefrontal cortex. So why did this happen? One compelling theory lies in the large groups which hunter-gatherer societies formed, and which we've inherited today. It's known as the social brain theory, and it's something I'm about to explore in a very personal way.
I've been working on these documentaries now for nearly 20 years. I've worked with all sorts of people. This is the first time I'm working with someone I know very well, my wife Julie. We've been together for over 40 years. I was asked to bring along either a good friend or my wife, and I don't have any good friends.
Yeah, not really.
You weren't completely happy to come along.
It's not my kind of thing.
You're not a show-off like me, is that?
No, I'm not a show-off like you. Not many people are show-off like you.
But anyway, I hope you know what you're letting yourself in for this time.
Not really, but I might finally get to find out what you do on these shoots.
We'll stay friendly and see what happens.
Okay.
Okay.
Julie and I have been offered an unusual opportunity. We've been invited to drop in on a major 5-year exploration of a unique aspect of the evolution of the human brain. Researchers from Nottingham Trent University have taken over an entire house. Cameras have been placed everywhere. And while we're here, every move will be filmed from different angles, and the footage will be analyzed by researchers hidden away in a different room.
Are you ready to play a game together?
Yeah.
For months now, psychological and behavioral tests have been carried out here with 120 pairs of best friends or couples.
The game that you're going to play is called Lost on the Moon.
Lost on the Moon is a puzzle game.
Do we need this?
Well, we've been asked to put these items in order of importance to help us survive on the moon.
Why would we need a box of matches?
Because there's no atmosphere on the
There's
moon,
no
so
atmosphere, so you can't light
have it light a
anything.
fire. I'm going to be hungry. I'm just saying I'm going to feel peckish and
Well,
thirsty.
Yeah, but it's not just about you, Jim.
Well, you probably want a snack.
What have you got?
I've got the pistols.
Oh, pistols.
What we're going to shoot?
You.
Apparently, we're going to need the pistol because if we run out of food, Julie's going to get hungry and she's going to want to shoot me.
I'm not going to want to shoot you because I might get hungry.
Just because you want to shoot me because I'm getting on your nerves. Fine. As far as we were concerned, this is all about, you know, whether we can survive on the moon until we're rescued. But it's clearly about, actually, whether we're cooperating.
We're quite good at that.
It might not seem like it all the time, but we actually do manage to compromise and cooperate.
What do you mean, doesn't seem like that?
Well, it just sorry, dear.
While all this is happening, our facial expressions are being recorded, and a team of evolutionary psychologists are analyzing our results. Then they call us for a meeting.
So we've been videoing you while you've been doing all of these tasks, and we've been measuring your facial movement, whether you furrow your brow, whether you raise your brow, whether you smile, whether you wrinkle your nose.
Funny look, isn't it?
Your eyebrow raises.
Eyebrow raises.
So this gives us a huge amount of very detailed information, of behavioural information that's happening during a normal conversation.
Jim has a rate of approximately 127 muscle movements per minute.
Is that good?
That's very high.
Quite high, yeah.
The average is about 100.
Right.
Julie had slightly less facial movements than me, but it might be because she tends to play her cards close to her chest.
I might just be one of those really deadpan people.
The poker face.
This project is investigating how much we use our facial expressions to bond with others, especially our nearest and dearest, and comparing it with other primates.
Many primates have facial expressions that are similar to ours, but our faces are the most expressive. And we think this sort of complexity is probably really important to humans as a species, and how we manage these really complex, intense social interactions we have with other people.
That's helped the evolution of our brains, the fact that we are able to communicate with facial expressions.
That's the basic idea behind the social brain theory, that what is difficult is the social stuff. So forming...
I'm Jim al-Khalili, a professor of theoretical physics. But today, my brain isn't pondering equations, it's being scanned.
You have about 160,000 kilometers of wiring in about 1.5 kilos. So enough to wrap around the Earth 4 times or take you halfway to the moon.
Goodness.
Like everybody else's, my brain contains about 100 billion neurons and over 100 trillion connections. That's more than the stars in the Milky Way.
This is what your head looks like inside.
So how did something so complex actually evolve? Spanning hundreds of millions of years, this story takes in the strangest of creatures.
So there's a tiny brain.
Yes, exactly.
Now, thanks to the latest scientific breakthroughs, we can tell the brain's extraordinary story like never before.
And that's the first whisper of a revolution, a cognitive revolution.
It's an epic tale. With amazing twists and turns.
Look how quick.
Oh, my word.
As we build artificial intelligence vying to overtake our brains, I want to find out what makes the thing in our skulls so special. This is my brain, and this is its 600 million year story. To try to understand the beginning of my brain's story, I'm snorkeling off the west coast of Italy. This is home to some very strange-looking animals. But go back some 560 million years, and you'd encounter the common ancestor of us and many of these creatures. It had a rather special shape. Probably a bit like this worm, with a head and tail and left-right symmetry. This animal was, like us, bilateral. But it had something else too. Many scientists now believe that this was the first animal to possess the basic building blocks of the brain. It was in an epoch known as the Ediacaran that these bilateria first emerged. To find out more about their early brains, I've come to meet some scientists who are working in these waters.
Okay, pull up now and we will collect everything.
They're pumping up the water from a depth of 10 liters up through this filter, which lets through all the tiny organisms that they don't want to catch, and it captures all the ones they want to study.
They're looking for zooplankton, animals like tiny worms that drift in water.
So this in here, it's hard to see, but this is zooplankton. We can analyze this under the microscope. Small algae will go through, and the zooplankton will stay in the filter.
Why is it studying this tiny worm helps us understand the ancient early evolution of the brain?
We have chosen to work on this worm because we were convinced that it has ancestral features, so that it would show us, for example, characteristics of the brain that are old, that have always been existing in animals.
Like the first bilateria, these zooplankton have a tiny cluster of nerve cells in their heads and a strand running down their bodies. These cells coordinate muscle movement and other organ behavior. And crucially, as well as coordinating, early brains evolved ways of sensing and responding to their environment.
And so it was. So the first thing we found about this worm is that it uses light-sensitive cells, photoreceptor cells, that it has in its brain. And these cells are the same to the ones that we have in our retina, the rods and cones. And this was a big surprise, a big finding, that these are the same photoreceptor cells.
So these are cells that are activated, that send signals to the brain when light impacts in them.
We have these in our eyes, so whenever we see light, these cells are activated and signal to the brain that there is light. And the very same cells exist in this worm.
Back on dry land, another member of the team shows me what these worms can tell us about early brains.
Aha! Here it is. So this is a young or juvenile.
Right. So how big is this? Is like maybe a millimeter or two?
This is 2 millimeters.
Okay.
And it has on the front a nice head. You can see the pharynx of the animal where they take up food. You can even see some movement inside these little dark dots, which are the eyes.
So it has 4 eyes?
It has 4 eyes. And you can see these dark speckles. These are the pigments of the eye.
Inside these simple eyes are molecules called opsins. When light hits opsins, they trigger an electrical signal which can make the worm move.
Can you show me how they react to light then?
Yes, so here you see the larvae swimming, and then we switch on a directional light stimulus on this side of the chamber. You see when the light comes on, and they start to move to the light.
Oh, I see. You're replacing sunlight with your artificial light and having the worm move towards it.
Yes, we provide this directional light cue here from the side.
The worm isn't seeing, thinking, or responding like we do, though. It has a simple nervous system that makes it move when light strikes its opsins.
What does that tell us about the early evolution of vision?
We think that the first eyes were just, in fact, very simple, a few cells able to tell the direction of light. And this kind of mechanism was probably there already in the common ancestor, last common ancestor of the bilaterian animals, around 540 million years ago.
At about the same time, other senses developed too. For instance, cells that could respond to nutrients, the forerunners of smell and taste. But these first bilateral animals also needed a way of sending sensory signals around their bodies to control muscles and organs. For that, they used a special type of cell, the animal world's fiber optic cable, a nerve cell, or neuron. It's taken over 100 years for scientists to understand the neuron. Much of that story happened just down the coast here in Italy, at a true landmark in the history of science. This is the Stazione Zoologica in Naples. It was founded in the 1870s by zoologist Anton Dorn.
Anton Dorn was an early and passionate adopter of Charles Darwin's theory of evolution by natural selection. And that passion drove him to create a station for scientists to study marine life. For his location, he chose Naples, because, well, Naples.
Dorn built this impressive laboratory right on the beach, with easy access to the Mediterranean and facilities to study all kinds of sea creatures. To explore how neurons and nervous systems worked, he had his team make some exquisitely detailed and beautiful illustrations. Dr. Graziano Fiorito showed me some examples.
There was a strong tradition by Dorn of supporting the study of various organisms.
Wonderful. So these are illustrations made for this journal, which is Fauna and Flora. 1896.
They were interested not only in the shape of the animal, the organs, but the nervous system, how the nervous system is organized.
Then, in 1932, here at the Stazione, a British scientist called John Zachariah Young, or JZ, made a breakthrough in understanding how the neuron works. Scientists had previously suspected electricity was involved in neuron signals, but these cells were always too small and thin to really study. Then JZ found an exception. Squid possesses neurons so long and thick, they're actually visible to the naked eye.
This is a classic video where JZ was showing how to dissect a squid. He's now removing some part of organs, the gills, in order to reveal this network, you see?
Right. JZ realized that because these neurons were so large, he could isolate one with the correct dissection techniques.
So these are all nerves?
Nerves.
Each one of those lines?
Yeah, and finally, this is the giant single nerve.
Scientists used electrodes to measure what was happening inside the neuron. And this led to the discovery of precisely how electrical signals traveled along it.
Would you say this was the beginning of modern neuroscience?
Yes, J.Z. Young published this paper in 1939, and this was the emergence, let's say, of neuroscience.
We now understand that nerve cells, or neurons, are the building blocks of all brains. Neurons take signals from molecules like light-sensitive opsins and transmit them through the body as electrical pulses. But how exactly do they do that? Most neurons consist of three basic parts. Dendrites, these branching filaments that pick up the signals, a cell body that processes the signal, and a long strand called an axon that carries a signal to other cells. But what's really incredible is just how fast the signal can move. Let me show you the patellar reflex, or the knee-jerk reaction. My director, Tim, has kindly volunteered here. So look what happens when I do this. Hitting the tendon triggers a nerve in the thigh, and a signal travels to the spine and back, and the thigh muscle jerks. Watch how fast it is. It takes about 18 milliseconds, which means the signal is traveling at about 200 kilometers an hour. Electricity doesn't flow along the whole length of an axon, as it might do in a copper wire. Instead, the signal starts when a tiny channel at the beginning of the axon opens, allowing electrically charged particles to flow through it. This then triggers other channels to open a little further along. It's rather like a domino effect. Each tile moves very little, but the ripple effect travels rapidly. Same in the axon. The charged particles themselves hardly move, but the signal races down. Ultimately, what we see is a pulse of electricity passing from one end to the other. And it all happens in the blink of an eye. But what makes the brain and nervous system so flexible is that neurons signal other neurons, creating a complex network. They communicate across a tiny gap called a synapse. Here, the electrical signal triggers the release of a chemical called a neurotransmitter, which crosses from one neuron to another. In this way, signals can cascade through your brain and body. And new technology means scientists can see and hear neurons at work. In Boston Harbor, on the eastern seaboard of the United States, a fresh haul of tasty crabs is just in. But before they reach the plate of a hungry diner, some of these crabs are being diverted for scientific research.
So what Karen's doing now is she's pinning out the sympathetic gastric nervous system.
Professor Eve Marder runs this lab at Brandeis University near Boston.
Once it's pinned down flat, she'll place recording electrodes on the nerves that will allow her to identify the neurons and record the motor patterns.
Eve's team are measuring electrical activity in the neurons.
These very fine white lines are the nerves themselves, and you can see how delicate they are.
And the technology here means those signals are converted into sound.
So I'm using the electrodes to record the activity of the nerves, and you can hear that the rhythm is going pretty fast right now. You're hearing that nice rhythm of derp, 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 derp. It's the circuit doing its thing.
This is a real-time representation of the crab's nervous system actually sending messages, much like the ones that control our bodily functions. But in an era of fear about climate change, with temperatures set to rise, the discovery also has a worrying aspect. Neurons are prone to fail as their surroundings warm up.
Every human neurological disorder is exacerbated by high temperature. The fact that poor people and ill people are less likely to be in environments where they can regulate their room temperatures and therefore their body temperatures means that we're potentially heading into a major health crisis.
This fragility means we should never take neurons for granted. After all, these cells carrying billions of pulses around our brains and bodies every second arguably make us who we are.
All the thoughts and all the dreams of the entire human race are just made up of these tiny electric signals racing along neurons. It's mind-boggling.
For neurons to become really effective and lead to brains like ours, they had to do more than simply carry signals. They had to take context into account, respond to stimuli in much more nuanced ways. To do that, evolution picked a group of ancient proteins and found a new use for them: neuropeptides. Let me give you a sense of the rather subtle ways these work.
You see, depending on circumstances, the same thing could be either good or to be avoided. Let me give you an example of how important this is even now.
Think about how you might respond to a delicious-looking cake.
If I haven't eaten for a while, then I really just want to tuck in. But if I've just had a big meal, then it looks a whole lot less appetizing. And if I've just been sick from food poisoning, then the same cake can look repulsive. Our brains have a way of adapting our behavior by reprogramming what the neurons tell us to do.
These subtle alterations of when and why neurons fire are controlled by neuropeptides. For instance, they change our behavior when we're with friends rather than strangers. Or how a mother acts when she's with her new baby.
All these new behaviors happen slowly over minutes or hours and are very different from the rapid response to, say, touching something hot.
Professor Corey Bargman at Rockefeller University in New York has made it her life's work to study how neuropeptides alter behavior and to understand how they evolved.
The neuropeptides are very old. This system is one of the first ways that the nervous system functioned, even before there was sort of a centralized brain, while it was still kind of fuzzy and distributed.
To understand neuropeptides, Corey studied a tiny worm known as C. elegans.
This is a single adult C. elegans, and here's a little adolescent next to her, moving around in real time. Because she's transparent, under the right circumstances, you can see every neuron in her nervous system.
C. elegans has precisely 302 neurons, and scientists have now mapped exactly how each one connects to the others. Corey's research examines how neuropeptides influence this basic nervous system and enable the worms to have complex behaviors, like socializing or courting.
Some of the worms were very interested in associating with other worms, and we can actually see these behaviors here in this movie. And what you can see is that they're tending to hang out together in these little groups. And this is an aggregation behavior. Aggregation behavior is very common in animals. Animals often associate with other animals in their same species. That could be called herding behavior in sheep, or it could be called schooling behavior in fish. And we see exactly this even in these very simple worms.
Normally, when worms bump into things, they move away. But Corey found that sometimes something very different happened. A neuropeptide you might have heard of called oxytocin was released, which changed how the neurons work and how the worms behave.
Oxytocin is required for mating behaviors in C. elegans. I'm showing you here a mating pair of a male C. elegans and its female-like partner. And in the male, there's a single neuron. It's this bright spot here that looks almost like an eye. That's the neuron that is making oxytocin when the animal is mating. And so this male is resetting his behaviors so that in the presence of a mating partner, he'll respond appropriately for this for the next 5 minutes. And then once she disappears again, he'll go back to the other kinds of exploratory behaviors that he's doing.
But what really surprised Corey was how neuropeptides like oxytocin changed behavior in almost all living things.
We found that there was an oxytocin in ants, which of course are a social species, and it's involved in some of their social behaviors as well.
And in mammals like us, oxytocin has developed a crucial role.
Oxytocin is released when a female mammal gives birth. It helps to rewire the mother's brain to stimulate bonding between the mother and her offspring. It's being able to flip from one state into another. And we all know that experience in our own lives. We all know how emotional states are changing our behaviors on a moment-to-moment basis.
In our brains, neuropeptides control our moods, bonding, love, anger, sex. In fact, modern drugs like Ozempic, which is set to revolutionize obesity, is linked to the action of the neuropeptide GLP-1, which controls appetite. It's quite incredible that so much of our behavior is based on these proteins inside our brains that evolved hundreds of millions of years ago. The next huge leap in the brain's development happened about 530 million years ago, in what's known as the Cambrian explosion. Evolution went into overdrive. There was a huge burst of new, strange-looking species. Big creatures with shells, thorns, fierce claws and hooks. And one particular adaptation that would have far-reaching consequences for our brains.
I'm about to show you something really exciting. Hi, Philomena.
Hi.
Can I have a look at
Yes.
what's in that dish? Thank you. I'll put it on this dark background. What's inside here is really rare. These long creatures, that's called Amphioxus. It's regarded to be very similar to the earliest, the common ancestor, about 500 million years ago, that marked the transition to the first vertebrates. Maybe we can have a closer look under the microscope.
Here we have the adult Amphioxus. Here we can see the anterior part, the head. Here, the posterior part, the tail.
And from head to tail, the nerve cord has been supplemented with a string of cartilage and bone, a primitive spine. This gave the ancient version of Amphioxus an advantage over earlier Bilateria. It would allow brains to get bigger.
So there's a tiny brain.
Yes, exactly. If we go more in, we can see here we have this enlargement, and here there is the brain of Amphioxus.
The rigidity of spinal cords and early skeletons allowed vertebrates and their brains to grow. Amphioxi have 20,000 neurons. You can get a sense of the advantages of having a smarter brain and internal spine and skeleton by watching this fish tank.
Right now, this looks like a perfectly peaceful habitat. Fish gently moving about, calmly, harmoniously. But that's because they're not thinking about their lunch. Let the feeding commence.
Vertebrates of all kinds proliferated in the Cambrian explosion. Thanks to their speed and maneuverability, the ones with spines and bigger brains were great at both hunting and escaping.
The main scan is basically ready.
At the MRI scanner at University College London, I'm being shown the debt my brain owes to my early vertebrate ancestors.
That's your spinal cord coming up into your brainstem. Brainstem is one of the sort of evolutionarily older parts of the brain that keeps your heart beating, your lungs pumping, move your eyes around. So lots of important things.
Another crucial component of vertebrate brains is the hippocampus, which we use for memory formation.
The hippocampus is a little C-like structure that shows up here. And I can see it because I'm used to looking at hippocampi, but it may be a little bit challenging. It's sitting just below your amygdala, which is part of the brain that's sort of an emotional center.
This new large vertebrate brain was matched by an increase in body complexity and size too. And this meant the brain and body needed more sophisticated internal communication.
Brains not only had to get better and better at sensing what was going on in the world outside, but also what was going on inside the body. And one very important way they do this is fascinating. They use a molecule that works a bit like a telegraph key sending Morse code. A telegraph key is pretty simple. A battery connects across a lever arm and a contact below. So when I press down on the arm, I complete the circuit, and a current flows through the wire, enabling me to send a message. In essence, a mechanical force, in this case, the pressure of my finger, is triggering an electrical signal.
For years, scientists suspected something similar was happening in our nervous system. Something was transforming mechanical force into some sort of signal to the brain. For instance, as a stomach fills, its walls stretch. And somehow the animal knew it didn't need to eat for a while. But how did this actually work? The answer was finally discovered in 2010 by Professor Ardem Pataputian at Scripps Research Institute in Southern California.
We're used to thinking about taste and smell as sensing chemicals, but this is actually sensing physics, right? It's mechanical forces, pressure. You have to turn this physical stimulus into an electrical or chemical signal that neurons would understand. And this transformation has been a big mystery.
The challenge was to find a molecule that, like a telegraph key, was triggered by mechanical pressure to send an electrical signal. So Ardem's team built an ingenious probe that gently applied pressure to cells just a few thousandths of a millimeter across.
We have a video here of the cell poking assay under high magnification. And so what we're doing here is using that to deliver submicrons, so very, very small fractions of a millimeter indentations to this cell that's sitting on this dish right here.
After nearly a year of poking and prodding, they finally found a single molecule on the cell's membrane which triggered a tiny electric current when pressed.
So when we poke very far into the cell, we see a very large current. And when we only poke the cell a little bit, we see a very small current, or none at all. And this tells us that the cells are really exquisite sensors of mechanical force.
They called this molecule piezo. This is a model of one of the molecules that Ardem and his team discovered, the biological world's telegraph key. It's embedded in the surface membrane of cells like the neuron. And it's tiny, just a few millionths of a millimeter across. But despite that, it's a really complicated molecule. It's a single protein made up of roughly 50,000 atoms arranged in this intricate shape, with 3 spiral arms and a cap in the middle. The shape of the protein is crucial. When the cell wall stretches, the whole structure flattens out, and the center opens up, creating a channel through which charged particles can flow into the cell. This is analogous to pressing the telegraph key, allowing an electric current to flow. We now know there are hundreds of thousands of piezo molecules throughout our bodies, signaling our brains. They can monitor our blood pressure, appetites, the need to wee, the timing of our heartbeats, and even regulate our breathing. And because hundreds of thousands of piezos are embedded in our skin too, they enable the most exquisite and underrated of all senses, the sense of touch. Think about it, our ability to touch is amazing. We can feel the tiniest breath on our fingertips, or the feel of a single grain of sugar, or the gentlest brush of a feather. Scientists say we can sense something on our fingertips that's just 1/500th of the width of a human hair. Having discovered piezo molecules, Ardem Pataputian's team then looked at where evolution had used them, and what they discovered was astonishing.
They seem to be pretty conserved across evolution. So we don't see them in bacteria, but in anything that has a nucleus, even unicellular organisms have them. We have seen them in flies, in worms. Plants, of course, have to sense mechanosensation as well. Roots, as they navigate through the soil and hit a harder surface, they have to decide to go through it, or if it's too hard, go around it.
The fact piezo molecules can be found across such a wide variety of life tells us they evolved a long time ago.
Our sense of pressure sensing that we mainly think of as sense of touch was present hundreds of millions of years ago in organisms that were just maybe swimming in the ocean and bumping into different things and needed to know what they were bumping against.
Up to this point, all life on Earth had lived and died in one place, in the oceans. The land was sterile and empty. But then things changed. Towards the end of the Cambrian explosion, about 500 million years ago, we think the first complex life began to creep out of the sea and onto the land.
Those first things that colonized the land were algae, mosses, basic bacteria. But then larger, more structured organisms began to emerge. Plants evolved root systems that went underground. Tall grasses emerged, and eventually trees.
Following them out of the water were early insects too, crawling and burrowing and wriggling things.
For sea creatures that lived close to the shoreline, the land now represented a new and potentially resource-rich environment. But of course, these animals would have to evolve considerably if they were going to survive above water. The process took millions of years.
You have to jump forward to 375 million years ago to see the first vertebrates starting to venture onto terra firma. This is a mudskipper. It's similar to the kinds of animals that were beginning to evolve at this time. Their eyes have moved upwards on their heads instead of on each side with most fish. Many scientists now believe that the way eyes worked out of the water led to one of the most significant turning points in the evolution of the brain. Professors Malcolm McIver and Dan Dombeck at Northwestern University in the States have been studying this critical moment in evolution.
With just the rising of the eyeball out of the water, you go from a medium which absorbs light so intensely that you only see a body length in front of you to essentially a perfectly transparent medium. That gives you a huge multiplication in your visual range.
Now, the key reason for that is to do with physics, or more specifically, the physics of light. You see, light travels far better through air than it does through water. That might sound rather obvious, but let me show you. Safety goggles on. What I have here is a tank full of seawater and a laser pointer that shines light through the water to be picked up by a detector on the other side, which I can read here.
When I turn on the laser, the light meter reading doubles from 25,000 to 50,000 lux.
That's how much light is getting through the seawater to the other side. Guys, give me a hand here. Okay, thanks, guys. Right, now we do this again.
We'll repeat the test, but with an empty tank. And the reading has jumped to over 100,000 lux. Air is at least 20 times more transparent than seawater. You can clearly see this when you swim in the sea. You just can't see very far underwater. That's because water is denser than air, and often murkier too because of mud or sea life. And this had huge implications.
As animals moved out of the sea and onto the land, the amount of raw visual information they could receive increased enormously, and their brains had to evolve to keep up.
How are you going to process all of that visual input to survive threats that are coming at you or to find food that might be trying to escape from you some distance away? So you need to have, you're going to have to up your computational brain in order to process all that information.
Malcolm and Dan believe that moving from sea to land led to a massive growth in size and power of the brain.
I want to demonstrate with these two skeletons just how big a change there was in relative brain size between ancestral fish and mammals. And so here is a fish that's in real life about this big, and its body mass a bit larger, but it's approximately similar to what a cat is. And yet, the total volume of this animal's brain is about 2 teaspoons, and it's a factor of 10 larger brain in mammals compared to fish.
The consequences of the move to land are stamped all over our brains.
Significant regions of the brain have evolved to make sense of the mass of visual data pouring in via optic nerves connected to our retinas. Regions like the primary and secondary visual cortex, the posterior parietal lobe, and the fusiform gyrus.
But though the move to land put down the foundations of these parts of my brain, it very nearly never got going, because around 250 million years ago, catastrophe struck. A massive series of volcanoes erupted in Siberia. The cataclysm exposed a huge geological fault stretching for hundreds of miles. And from this gash poured billions of tons of hot lava. Some suggest this flooded the plains below and came into contact with one of the largest exposed coal deposits in the world.
Picture the scene 252 million years ago. Billions of tons of red-hot lava, billions of tons of thick, black, dirty coal deposits. The result was a toxic gas release on a catastrophic scale. Poisons like sulfur dioxide, nitrogen sulfide, and methanes filled the atmosphere. They then mixed with moisture in the air to create powerful acids, which then rained down, wiping out nearly all life from the face of the Earth.
As well as this, the volcanoes belched carbon dioxide from deep inside the Earth. Trillions of tons, suffocating almost all plants and animals.
Today, we consider the world to be on the brink of a carbon dioxide catastrophe. There are 420 parts per million of carbon dioxide in the atmosphere and rising. But 250 million years ago, the Siberian volcanoes released 5,000 parts per million, over 10 times more. There was total devastation. 95% of all species were wiped out. In the whole history of life on Earth, it's the closest we've come to total extinction.
But of course, it wasn't the end. Some things did make it through.
The survivors were smaller animals that could hide away, they could burrow underground, they could crawl into crevices or slither into the mud. They could survive on less oxygen. They could cope with the heat, the darkness, and the toxic chemical mix. The scene was now set for one of life's most incredible comebacks, and one of the most profound moments in the story of our brains.
This is the Vale of Glamorgan in Wales. After the massive extinction event, this whole area was an archipelago of small, low-lying, linked islands, which makes it particularly interesting to paleontologist Dr. Ellis Newham.
What was this place like back then?
It's 50 million years after the end Permian mass extinction, the Great Dying. So life was bouncing back by then.
We're now firmly in the era of the first dinosaurs, the gigantic creatures that were coming to dominate the world. But Ellis is more interested in a much, much tinier creature, one whose remains literally fell through the cracks.
Within these islands, there was cracks and fissures that naturally developed. And so any animal that died, their remains would have got washed in. And so this gives us an amazing snapshot onto what life would have been like on those islands 200 million years ago.
And what Ellis and his colleagues found deep in these rocks were new clues to how our brains were actually formed. Bones from a tiny rat-like creature called Morganucodon, or Morgie.
We found thousands of isolated teeth, jaws, and bones. And I brought one here with me to show you.
I mean, it's tiny. This ain't no T-Rex. So what did this creature look like?
It was about the size of a shrew. It might have been furry. It probably had whiskers. And it was kind of scrabbling around the undergrowth in what is now South Wales.
Right.
So it's definitely insectivorous. It fed on beetles and kind of gnatwings and things like that were living with it at the time.
But what no one expected is what they saw when they started looking inside Morgie's skull.
Colleagues CT scanned one of these skulls, and what they found was pretty amazing. So this is a cast, a 3D print of a model that they created of the brain of Morgan Euclidon.
Wow. Very tiny.
Absolutely tiny. Maybe I'll get a scaled version for you.
Right.
So here we go.
On top of Morgie's brain, they noticed an intriguing new development.
We started noticing this thing. You can see this lumpy bit here.
And so the brains of other creatures that lived around that time didn't have this bump?
As far as we know. So Morgie shows a kind of a jump step in the development of the brain size and also the brain complexity.
This bump is a new part of the brain called the neocortex, and it's what sets creatures like Morgie apart from other animals of the time.
It's a small bump with huge implications because it's the bump that makes our brains so unique. So where did it come from?
To find out more about the neocortex, I've come to meet Professor Steve Brassate, an expert in paleontology and evolution.
This is Morgie's brain. This is a digital model from a CAT scan. And just to orient ourselves, this bit here at the front, that's the olfactory bulb. That's the part that controls our sense of smell. Back here is the cerebrum, one of the main components of the brain. And up there on the top is that curious little bump.
And that's what I'm interested in. So where did that come from?
We have a few clues that we think helps tell that story. And the first one of those is if we look at the bones of Morgie.
Unlike any animals before this time, the microscope reveals Morgie's bones are honeycombed by tiny holes, evidence of a massive step change in blood flow.
Those are just digital renderings of the channels for blood vessels. And the key thing is that their bones, their skeletons, were becoming much more full of blood and blood vessels. And that means that they could have higher metabolic rates.
So their bodies were just working harder?
Their bodies were working harder, they could grow faster, their metabolism was increasing. That's part of the story. But there's a second clue to this as well, which we'll have to take a look over here.
Okay.
And this is a little fossil discovered about a decade ago by teams from here at the National Museum of Scotland, out on the Isle of Skye. And if we put it under the scope, we can see what it is. The bit sticking up from the top, that tells us what this fossil is. And those things are teeth. So this is a jawbone. But when you blow it up in size, you 3D print it, you can see all of these teeth here, and it's in these teeth that tell us the importance of these fossils. And they tell us a few things. These teeth, they're really sharp. There's lots of little points. And when the teeth came together, all those points would interlock, and they would bite really strongly, be able to chew the food. But the other thing is, the babies did something different. We can tell that these animals had one set of baby teeth, one set of adult teeth. And that means that the babies were drinking milk.
Okay. What does that tell us?
So these, of course, are mammals, just like us. These are early ancestors of ours. What we see with these fossils at this time, these mammals were entering an age of gluttony.
So these mammals ate more, their offspring drank milk, and they had more blood flow in their bones. This wasn't just the start of mammals. It was a new way of being.
That tells us the big reveal here, and that is that we think that these mammals were becoming warm-blooded. And that means you have a constant body temperature, a high body temperature, and your body temperature is high and constant regardless of the season, regardless of the time of day, regardless of where you live. And that's a very special superpower. That's what we have.
Warm-bloodedness enabled animals to exploit new niches, hunting at night and in the cold.
So it takes resources to be warm-blooded. If you think about the cost of heating our homes, it's expensive.
A mammal needs to eat about 8 times, give or take, as much food as a cold-blooded animal of a similar size.
So bringing this back to the beginning, how does all these changes affect the brain of Morganucodon?
That's where that bump on the brain comes in. And that's the neocortex. It's clearly getting larger. That's the expansion of the brain that's happening at the exact same time in evolution as these changes to the teeth, changes to the diet, changes to the metabolism.
So you have the evidence, the bones, the teeth, suggesting they are eating more, they're metabolizing more, they have to find food more often, and to do that, they need bigger brains.
That's it. That's the story, and it's a huge story, and it's one of the biggest changes that we see in the evolution of animals.
So what did these new parts of the brain allow animals to do? It seems to have led to the development of a remarkable new ability, a true superpower.
That superpower is what we might loosely call imagination, our remarkable ability to remember scenes from our past or to conjure up ones in our future. Cast your mind back to when you were a young child.
Try to remember what it was like.
Now conjure up your next Christmas or family gathering.
Can you see in your mind's eye what it will be like?
What's amazing is how effective this internal visualization can be, our ability to see the past or imagine the future. We can link it to smell and even to our emotions.
To investigate the mammalian origins of imagination, Malcolm McIver and Dan Dombeck built this, an open maze with food at several reward ports. And because genuine early mammals no longer exist, his team used mice to see how they visualize the world around them. And to help motivate the mouse, an unusual predator.
We came up with a large arena where we could essentially run a predator-prey scenario, but with the predator being an autonomous robot that we have designed and developed. The robot doesn't physically attack the rodent in the space, it simply puffs an intense blast of air. And rodents really dislike that, so they avoid the robot.
The team then attached probes into the mouse's hippocampus, which processes location information from the mammalian neocortex.
And we asked what exactly is going through the mind of the mouse. Is it imagining a future position of the robot? Is it imagining a future position of itself?
They put the mice through the maze thousands of times, an environment specially designed to make them think ahead. They then took data from the probes to get a sense of what was happening inside their brains.
So here's the experiment that you saw earlier. We've also included neural data.
The blue dot represents where the mouse actually is, and the red dot is where the robot is. But incredibly, by reading the signals from the mouse's neurons, the team could tell where the mouse was planning to go. That's the pixelated blue blob. Malcolm believes this reveals important clues about the mouse's imagination.
You can see that essentially we have the mouse thinking about where it's getting to in the next little bit. It's actually here, and this is where it's going to be soon, and it seems to be thinking about where it might be in the future. So right now, the mouse imagination of its location and its actual location are very close.
But then something remarkable happens. The pixelated blue blob moves away from the blue dot. This suggests that the mouse is thinking of where to go next to find food.
Right here, it skips over to here, And it starts to coalesce around this next reward port, which you'll see in a second is where it's going to go to next. So it appears to be thinking about, this is a reward port, and it appears to be thinking about the next reward port it's going to be visiting. And that is really interesting, because watch what happens after the mouse leaves this position, after it's checked that reward port. It actually goes over to exactly the location it seems to have been thinking about. So it's thinking of the future, it's imagining the future, which is exactly what we'd hope to see with these more sophisticated behaviors that the arena is exhibiting.
This is a bold claim, that leaving water for land, where long-range vision makes planning a matter of life or death, may have led to the ability to imagine.
If you're going to do the more complicated thing, which is essentially representing the future and future opportunities and threats, that requires a lot of neural power. It's a massively intensive task. So this is one of the demands on the nervous system that presumably feeds into this huge 10x increase in brain size in the mammals and in the birds relative to ancestral aquatic organisms.
Being able to visualize the future, to plan and solve problems, powers enabled by the burgeoning neocortex, would eventually come to give mammals a huge evolutionary advantage.
They survive, probably in large part because of their large brains, their high intelligence, their ability to solve problems. Within a few 100,000 years, we start seeing mammals the size of pigs. Within about 1 million years, we start to see mammals the size of cows. So as these mammals themselves are getting larger, the brains are getting larger, but you can see it better on CAT scans. These are the brains, you can kind of see them getting bigger, and that gray bit at the top, that's the same thing as the bump in Morganucodon. That's the neocortex, but you can see it's getting bigger and bigger and bigger. And as mammals continue to adapt and evolve into dogs and cats and bats, whales, elephants, kangaroos, all the mammals that we know, monkeys, apes, and eventually humans, that neocortex, it came to envelop the surface of the brain.
And so most of what we regard as the brains of humans actually emerge from that bump in Morgie.
Exactly. And that's the first whisper of a revolution, a cognitive revolution. And we see in ourselves that same structure, but at just much grander scale.
So how important would you say was the emergence of warm-blooded animals in the evolution of the brain?
To me, that was the single biggest Big Bang instance in the history of brain evolution. So much about the brains that we have today was shaped by our metabolism being evolved by our fossil ancestors.
If Malcolm McIver, Dan Dombeck, and Steve Brusate are right, then the movement to land and the development of warm-bloodedness are responsible for the single largest neural growth spurt in 600 million years.
It gave us the greatest gift of all, the brain.
Next time, foraging, tool building, child rearing, and relationships. How our social lives have shaped the human brain. Curious about how MRI technology can reveal the secrets of your brain? To watch the Open University's new animation exploring how an MRI works and what it reveals, scan the QR code or go to bbc.co.uk/secrets of the brain and follow the links to the Open University. I'm Jim al-Khalili, a professor of theoretical physics, and I'm exploring how the most complex objects we know of in the universe, our brains, evolved.
This is what your head looks like inside.
In the last episode, we went from the first neurons to the early mammal brain. Wow. In this episode, I learn how life in the forests shaped our brains, and how our primate ancestors solved problems to survive.
Look how quick!
Oh my word!
With my wife, I investigate how relationships made us intelligent.
You're not show-off like me, is that?
No, I'm not show-off like you. Not many people are.
It's a long evening.
And how our brains are wired to be social.
The single best predictor of how long you're going to live into the future, it's simply the number and quality of close friendships.
As we build artificial intelligence vying to overtake our brains, I want to find out what makes the thing in our skulls so special.
This is my brain, and this is its 600 million year story.
"66 million years ago, the Earth lay in desolation and darkness. An asteroid smashed into the surface of the planet. Soot and ash and vaporized bedrock dimming the sun. Perhaps 3/4 of all life on Earth was wiped out, including the gigantic dinosaurs which had ruled the world. But as the Earth slowly recovered, forests returned and spread across the land. And with the dinosaurs gone, small creatures colonized this new habitat. Among them were our ancestors. Now, here's the mystery. How did they survive this catastrophe? Dr. Ornella Bertrand studies the extremely limited evidence that survives from this dark period in the Earth's history. That is not an easy job. You've drawn the short straw when it comes to paleontology, right?
Definitely, because it's very difficult. There's just not that many fossils. We find teeth, we find like a bit of bone sometimes. One group that I'm really interested in understanding is a group called Plesiadapiforms. They were little like warm-blooded mammals.
Plesiadapiforms resembled modern shrews. It's thought they used their sharp claws to cling onto the branches of trees in the primeval forest. They're the closest we've found to the predecessors of all today's primates, including us. Ornella studies their skulls, but she doesn't have many to work with.
We really have like 6 good specimens so far.
6 specimens in total
of
these creatures?
Yeah.
Right.
So that's very little, but we can learn so much just from those different skulls.
Ornella has a 3D print of the skull of a specimen called Ignatius grey-bullianus, upscaled so we can see the details. By scanning the cavity inside the skull, she's been able to produce a cast of the missing brain.
Ta-da! It's amazing, isn't it?
It is incredible.
It gives us like so much information about what actually those animals were able to do. And for example, the structures in front.
I was just going to ask you there.
Yeah.
Funny little knobbly bits at the front.
Yes. We still have those structures, the olfactory bulbs in humans.
They're olfactory. Okay, so that's for smell. For the
Why not? Yeah, exactly.
Plesiadapiforms had very large smell processing systems. Ornella believes they may have scavenged at dusk and at night when a strong sense of smell is very useful.
I imagine them being like quietly moving among the branches. That's kind of, how I see them.
For this documentary, I've had my brain scanned, and I've been carrying around a 3D printed version of my brain.
Wow.
Which is here, which I'm very proud of.
I would be, that's amazing.
Which we can put alongside Ignatius. I'm not boasting. There's been some evolution.
So, just a little bit.
I'm interested in a particular part of the brain, the neocortex.
So here we have the neocortex of Ignatius. It's very small. It's actually just this part of the brain.
In Ignatius, it covers about 20% of the top of the brain.
That's very different in a human brain, isn't it?
Very, very different. You can see like the neocortex in humans actually is covering the entire rest of the brain. This is amazing how big that it got in humans.
A rudimentary neocortex helped early mammals analyze the world around them and respond to it. For humans, it's become the seat of advanced thinking, reasoning, and perception. But the mystery is, why did our neocortex grow so large compared to any other mammal? There's a compelling theory, and it's to do with climate change. About 56 million years ago, greenhouse gases released by volcanic activity caused sudden global warming of up to 5 degrees. It was the beginning of the Eocene era, a time of astonishing changes.
In the hothouse environment of the Eocene, dense rainforests spread around the world. They even reached high enough latitudes to be within what is today the Arctic Circle. And as with the rainforests of today, life proliferated.
Just look at these extraordinary fossils found near Frankfurt in Germany, dating nearly 50 million years ago. They give us an incredible insight into the competition which raged in the forests of the early Eocene. There were birds and agile tree-dwelling rodents. There were highly dangerous predators too.
All of this meant fierce competition for food and resources. And with all these new predators around, it was much more dangerous too. Only those creatures able to adapt would survive.
In these changed circumstances, simply staying alive would take brains as well as brawn. As I'm very quickly finding out, a tree is a tricky place to call home.
It's actually quite difficult moving around up here. It doesn't just take physical strength. You need judgment. You need to decide whether a branch is strong enough to hold your weight, whether it's too wobbly. You need to plan a route through the trees.
On top of the physical and mental agility you need, coping with the incredibly complex visual field of a canopy of leaves poses yet more problems.
I'm finding it really difficult peering through the leaves and twigs and branches. Keeping an eye out for predators, as well as foraging for food, must have been a real challenge.
In the face of these evolutionary pressures, a new kind of creature appears in the fossil record of the Eocene. They're called crown primates, the forerunners of all today's primates, including, of course, us.
Is this the way we sit? Like this?
Early primates were well adapted to life in the trees.
What's interesting is the way he's grasping the branch.
During the Eocene, primates evolved gripping hands and feet with nails.
They're much more dexterous. They're able to hold on to this branch. It also means they can use these hands to find food, to forage, to manipulate objects much more easily.
The other thing to notice is that they have eyes at the fronts of their heads, not on the side. Animals with eyes on the side of their heads have a wider field of view and can spot predators better. But eyes at the front help with what's ahead of you, judging the distance to the next branch or locating food. The effect of millions of years of tree dwelling is clear when we look at the fossil record. This is the brain cast of Ignatius, the Plesiadapiform we've already seen, clambering around the forest in the years after the dinosaur extinction. And this is the brain cast of a primate called Runeia. It evolved around 20 million years after Ignatius. With Ignatius, the neocortex covered 20% of its brain. With Runeia, it now covers half its brain. The neocortex analyzes visual data. And it has many other functions besides. It works in extraordinarily complex ways, which we take for granted in our everyday lives.
Take, for instance, what happens inside our brains when we do something as deceptively simple as reaching out and grabbing an apple. It's actually far more complicated than you might think.
I'm getting an extraordinary insight into what's involved when we reach out and grab something. Dr. David Pitcher at the University of York has carried out a functional MRI, which measures the way the blood flows in my brain.
Okay, Jim, this is going to be an 8-minute scan.
While he plays me videos of reaching out and grasping an apple.
My colleague, Amel, is going to put this
hello, right
on your head.
Yeah, I think so.
Then he uploads my results to this machine, which uses spatial recognition technology to guide me around my own brain.
Now where she points to with that pointer, this is what your head looks like inside.
Lovely death mask as well, isn't it?
Yeah, oh, I can rotate that for you as well, so we can see.
My scan shows astonishing activity across my cerebral cortex as I watch the video. First, David shows me how my visual processing works, a complex system which really started to develop with those early primates.
Light hits your eyeball, that's then converted into neuronal signaling. That information is then sent down the optic nerve, all the way to primary visual cortex, which in humans is right at the back of your head.
As I was looking at the apple, my visual cortex connected with another part of my brain, which helps us recognize objects.
You see this pathway that lights up. This is the ventral object recognition pathway.
And that pathway leads to the bit of the brain where I'm storing information of what apples look like.
Yeah.
The hard drive of my brain.
That's where we store everything, yeah.
Then a second connection activates from the visual cortex at the back of the brain to the parietal lobe. The parietal lobe judges where things are in space around us and where our body is positioned. But to issue the orders to the body to move, you need a third part of the neocortex, the primary motor cortex.
This is your motor cortex, and there's parts that control all the motor function in your body. And if Amel moves the pen down the side of your head, we're sort of tracking through different parts of the motor cortex. And where we come to sort of roughly where Amel is now, that's roughly where the hand area is. So that's the...
So different parts of the motor cortex physically relate to different parts of my body.
Exactly.
The visual, parietal, and motor cortex all developed in our primate ancestors. And a new area of the neocortex was also beginning to evolve. It would become the most sophisticated cognitive area of our brains.
The other thing you have to do is be able to plan and control the motor action. So if Amel brings the pointer forward a little bit, is the prefrontal cortex. It's where we do all our thinking. It's where cognitive control happens, the voice inside your head.
So just picking up an apple means the visual cortex, the parietal lobe, the motor cortex, and the prefrontal cortex all have to work together.
So 4 different parts of the brain are all involved in the simple action of reaching out, picking up an apple. Visual cortex, parietal lobe, motor cortex, frontal cortex.
From 40 million years ago, the creatures we now call monkeys were evolving. Their neocortex was becoming bigger and more complex in structure. In today's primates, the larger the brain, the denser and more closely packed the neurons, the more networks they develop, the cleverer they become. So what were the driving factors which caused some primates to evolve differently from others? And why did humans evolve the most intelligence? One theory has to do with the ability to find the best, most calorific food. And it's something we can study by looking at 2 species of modern monkeys side by side.
So this is the viewing deck, and here we can see both species, the capuchins and the squirrel monkeys. There are relatives, but if we study enough of our primate relatives, we can start to make inferences about what our ancestors, our primate ancestors, might have been like.
This is the Living Link Centre at Edinburgh Zoo. Professor Amanda Seed studies these species side by side to try to understand the story of our own brains.
The capuchin monkeys are our larger monkeys. They're more robust. They have this characteristic dark colouring on their head and the little tufts at the front. The squirrel monkeys, on the other hand, they're smaller, they're more slender, look a little bit like they've been dipped in chocolate.
Capuchin monkeys and squirrel monkeys evolved together in the same place, the forests of the New World. And yet, capuchins have a much larger neocortex compared to their body size and exhibit greater intelligence. The question is why? And what might that tell us about the evolution of our own brains? Amanda and her staff have devised a challenge these monkeys haven't encountered before. They've hidden food in papier-mâché boxes. The challenge is which species will figure out that there's food inside and manage to get it.
Straight in.
Thank you very much.
Straight away, the capuchins have come out. They're very, very curious, and their first thought is, can I break it? Is there food inside?
Meanwhile, the squirrel monkeys hang around on the sides, and not one of them looks inside a box. Instead, they wait for the capuchins to finish feasting, and they pick up scraps from their table. Amanda believes today's test has helped confirm a theory which links intelligence to diet.
The capuchins rely on fruit to a much greater extent than the squirrel monkeys. The squirrel monkeys eat fruit as well, but their diet is largely comprised of insects.
The squirrel monkeys just have to wait for an insect to come along and grab it, and that happens a lot. But fruit only appears a few months every year. It's found around the forest in patches, in often hard-to-find places. So in order to find it, you have to remember and plan ahead.
An ability to map your environment in space and even time, to be able to predict when those resources are going to become available, now becomes useful. And so now we have more selective pressure for areas of the brain that can deal with that.
The penny's starting to drop for me now, that it's not so much that they have larger brains, they're smarter, therefore they're able to figure out things like, how to find food. The need to look for food has stimulated the growth of a larger brain over millions of years.
Over millions of years.
So it may be that at some point in our evolution, our ancestors were like the capuchins. They concentrated more and more on fruit, high-calorie fuel for growing brains. But it takes more intelligence to find. By 25 million years ago, the first apes were evolving. Ape brains are generally larger than monkeys, and they have more complex structures and networks. It's now thought that around this time, our ancestors took their first baby steps towards one of our most important cognitive abilities: complex spoken language. I've come to see something very unexpected, which might provide a vital clue. These gorillas live at Port Lympne Safari Park in Kent. I'm with Professor Gillian Forrester, who's been studying the primates at this sanctuary for 20 years.
I really like working here at Port Lympne because it feels like more of a collaboration with the gorillas. We work together, but they're in an environment that really suits them. The enclosure was made for them, not for people to view them.
Right. It's lunchtime, and the gorillas have an unusual treat. Nettles, which they find very tasty. they found a clever strategy to deal with their stinging leaves. They strip the nettles off the stem in one direction, a direction which doesn't trigger the spines so they don't get stung. One gorilla decides to literally walk away with her spoils.
Look at her go.
Fantastic.
We follow this gorilla to the inside area, where we catch a glimpse of a more complex form of nettle preparation, rolling the leaves into a ball to crush the stinging spines. This method of preparing nettles has been recorded in many different gorilla groups.
And you want me to try this, don't you? Yeah. Okay, right. Let's see how we go.
Let's see.
Well, okay. There we go. I was so careful not to get stung, which I think I've survived, that I've dropped the leaves.
Drop the leaves, yeah. Not as delicate as you, but nearly there. Yeah. I think it's interesting because eating the nettles requires the gorillas to take certain actions in order to not get stung.
Okay.
We could think of that as almost like what we'd call a syntax in language. When I speak, these are motor action sequences. So my mouth has to make the right words at the right time in order for you to understand what I'm saying. This is a syntax in language, and this is a syntax in its physical form.
Gillian has created some puzzle boards which require an understanding of more complicated syntax than nettle stripping. The boards have a series of cogs. The question is, can great apes turn them in the right direction, in the right order, to solve the puzzles?
Sorry, Jim, but you've got it the wrong way around.
Oh!
We're trying out her puzzle boards on two orangutan brothers, Malu and Hadi.
Oh, here they are.
Oh,
yeah. Oh, they're right in there.
Right away.
They've spotted the nuts, which Gillian has put at the top of the puzzle boards. But to get them out, they'll have to maneuver the nuts to the bottom of the board. On his board, Hadi tries using a twig as a tool, but that's not going to work. Over on the other board, Malu lands on the correct solution.
Oh, look at that.
Okay, that's Malu.
Wow.
Malu's worked that out.
Well done,
Here we go. Malu's got into the second set of cogs. And now he has, oh, look how quick.
Oh, my word.
I can't believe how fluid his movements are. He's getting to the last stage now. This is the double cog, the exciting bit. Will he think about, yes, he's thought about which way to turn it. He's gone to the access point. This could be the solution. Okay, he's been...
The last, look at the last stage.
No.
A big
so
brother has taken over.
Big brother's taken over, and Hadia's gonna get the reward.
But between them, they've solved
that's kind
it.
of yes, it was a collaboration.
Yeah.
Gillian is convinced that our ape ancestors developed the ability to solve ever more complicated physical syntax problems, and were unknowingly building the foundations of what would one day become language. Our ancestors split from orangutans around 17 million years ago, and from gorillas about 10 million years ago. Again, climate change was bringing new threats. Millions of years of slow global cooling meant that rainforests were retreating to be replaced by open grasslands and woodland.
Competition for the most nutritious and calorific foods in the rainforest was becoming ever more intense. Life for our primate ancestors was getting harder and harder.
Survival would now depend on making finely balanced judgments based on limited information about where to find food. To find out what mark climate change made on our brains, I'm visiting the chimpanzees who live at Budongo Research Unit at Edinburgh Zoo. Along with bonobos, they're our closest primate relatives. I've been told I can play a game with them to test their cognitive powers.
Not one of you said thank you yet.
So I head to the indoor part of their living quarters.
When the chimps start making their noises, it is really loud in here.
The chimpanzees have complete freedom to do as they like here, so I have to wait to see if one of them wants to join me. Eventually, 4-year-old Masindi decides she wants to play.
Masindi, look.
To get the game started, I show her 2 empty cups.
Nothing.
Then I cover them with a screen and put a grape under one of them.
Do you feel lucky? I'll put it under this one. Pretend to put it under this one.
She doesn't know which cup has the grape, so she guesses the cup on the left.
That one, okay.
But now I complicate things.
I move this cup back.
I remove the cup she doesn't want and offer her an alternative, a half grape.
Now, what do you want?
So now she has a choice. Pick up a cup which may or may not have a grape under it, or settle for a guaranteed half grape.
This one. That one you want. Okay, well, look what you could have won. But unfortunately, you have a half.
She's chosen the half grape. It's a safe decision, but she's missed the chance of winning a full one.
And that's the choice they have to make, whether they want half a grape for sure or a whole grape half of the time.
We play the game again, and this time she sticks with the cup.
You're going for it, are you? Are you confident? Clever.
Of course, Masindi's having to guess if the cup has a grape under it. But now, what happens if I give her more information to act on? I offer her 2 cups with 1 grape, as usual. But this time, I show her that one of them is empty.
Let's see, this is going to be the clincher. What are you going to... Good girl, yes, you got it. Well done.
She immediately realizes that the other cup must be hiding the grape.
She knew there was definitely a grape under this one, so choice between that and the half grape, she's sticking with the cup because she knows there's a grape under it.
The chimps also play video games, which simulate complicated foraging tasks, looking for hidden fruit scattered around a complex landscape. The game is helping investigate the theory that as the rainforests retreated, the need to find scarcer food required greater brain power, memory, and planning ahead. These games are posing a vital question. Are the chimps able to keep track of their own awareness of the world? What they know and what they don't know? The ability to keep track of what you know and don't know is a vital and mysterious part of human intelligence known as metacognition. It's something which particularly interests the head of the research unit, Professor Josep Call.
Metacognition is the ability to monitor and control your own thought processes, your own mental content. There is a thing called in metacognition that is called the tip of the tongue phenomena. When somebody asks you a question and you say, I cannot give you the answer, but I know that I know this answer. I just
all the time.
Yes, there you go. So this is metacognition, where even though you cannot produce the answer, you know that you know the answer.
One theory states that our ancestors developed metacognition as they solved problems posed by the environment, such as finding fruit in changing landscapes. It's thought this led to what's known as theory of mind. The awareness not just of your own mind, but others' minds too.
It's about knowing about your own processes, but also imputing processes to others. What others can see, what others want, what others intend, what others believe.
Thinking about others is a crucial step towards becoming the socially connected creatures which human beings are today. Our ancestors split from the lineage of chimpanzees around 6 to 8 million years ago. After that came a huge change in lifestyle, a change with astonishing consequences.
No one knows exactly how, why, or when it happened, but at some point during the millions of years after our own lineage split from that of chimpanzees and bonobos, our ancestors left the forests to make a new life out on the open plains. They became bipedal, walking up on 2 legs, so better able to adapt to different terrains and environments.
Between 2 and 3 million years ago, Homo habilis was evolving, the first human species. They were developing areas of the neocortex involved in planning, thinking, problem solving. And we start to see some astonishing results of improved brain power. Dr. Nada Kreischer is an experimental archaeologist. She works at the Ancient Technology Center in Dorset. I'm meeting up again with Gillian Forrester. She and Nada have both investigated the fascinating links between tools and the story of our brain.
So, Jim, I'm really excited to introduce you to Nada. She works in this beautiful place, and she actually teaches people how to make these ancient tool sets.
Wow. Okay, teach me.
So we'll just start with a brief history of some of the ancient types of stone tools that were made by our hominid ancestors. So this is a core, a flint core, where sharp pieces, as you can see down on the floor here, have been knocked off, and these sharp pieces were used as tools for cutting.
I want to know how much brain power it takes to create the kind of tools which our ancestors made. So Nada sets me a task to make this half-finished hand axe thinner.
So yeah, you can see that that's not coming off.
It's not brute force.
It's not just brute forced, unfortunately. Oh, he's breaking the hammerstone.
Oh, so he's actually broken the hammerstone and not the actual flint.
Which is quite an achievement, really. So as you can see, just hitting it as hard as you can up directly on the spot which you want to remove.
Is it working?
Isn't the right approach, really.
Okay.
Making the flint break in the direction you want it to turns out to be extremely complicated.
It might seem counterintuitive, but you actually have to hit it from this side to be able to fin it effectively. And before we do that even, we need to prepare a place for us to hit, to take a piece which should travel down there and thin the flint. If I do it right, I just need to do a few more preparatory flakes.
Only after a few minutes of careful preparation can she do this.
I'm going to hit on the top, and that's taken a piece which hasn't travelled as far as I want, but is kind of isolated this spot a bit more, so made it stick out a bit more. A lot of times when people think of stone tools, they're thinking of cavemen, they're thinking of mighty muscles, big hard hit, but it's also about planning and thinking ahead.
Gillian believes this supports the theory that our ancestors were using cognitive abilities which had developed millions of years beforehand. As we've seen, our shared primate ancestors had likely already developed an ability to master syntactical problems.
We've looked at nettle processing with the gorillas. We've looked... right down the middle here is your motor cortex. This is telling your body to move effectively. The hand area and the mouth area, they're really quite close to one another. And just in front of that area here is a region that we call Broca's area or Broca's region. It's always been thought to be a language-specific region. It is activated when we speak. But interestingly, it is also activated when a deaf signer signs their language. You're going to purse your lips when you've got a tight space, and you're going to maybe open your lips more when you're grabbing larger objects. This could possibly date all the way back to just feeding behavior. If you're going to pick up something small, you put it in. If you're picking up something big, you need to open your mouth wider. We also have this evolutionary theory that we might have been first speaking with our hands before language moved to our mouths. Only a small portion of the way we communicate is actually through our vocalizations. We're still communicating a huge amount about what we think and feel and intend through our facial expressions, our body postures, and the way we move.
So the theory is that carrying out complex tasks like tool making helped put in place the final bit of the brain which will be needed for language. Over the past 2 million years, our ancestors' brains tripled in size, particularly in the advanced cognitive areas such as the prefrontal cortex. So why did this happen? One compelling theory lies in the large groups which hunter-gatherer societies formed, and which we've inherited today. It's known as the social brain theory, and it's something I'm about to explore in a very personal way.
I've been working on these documentaries now for nearly 20 years. I've worked with all sorts of people. This is the first time I'm working with someone I know very well, my wife Julie. We've been together for over 40 years. I was asked to bring along either a good friend or my wife, and I don't have any good friends.
Yeah, not really.
You weren't completely happy to come along.
It's not my kind of thing.
You're not a show-off like me, is that?
No, I'm not a show-off like you. Not many people are show-off like you.
But anyway, I hope you know what you're letting yourself in for this time.
Not really, but I might finally get to find out what you do on these shoots.
We'll stay friendly and see what happens.
Okay.
Okay.
Julie and I have been offered an unusual opportunity. We've been invited to drop in on a major 5-year exploration of a unique aspect of the evolution of the human brain. Researchers from Nottingham Trent University have taken over an entire house. Cameras have been placed everywhere. And while we're here, every move will be filmed from different angles, and the footage will be analyzed by researchers hidden away in a different room.
Are you ready to play a game together?
Yeah.
For months now, psychological and behavioral tests have been carried out here with 120 pairs of best friends or couples.
The game that you're going to play is called Lost on the Moon.
Lost on the Moon is a puzzle game.
Do we need this?
Well, we've been asked to put these items in order of importance to help us survive on the moon.
Why would we need a box of matches?
Because there's no atmosphere on the
There's
moon,
no
so
atmosphere, so you can't light
have it
anything.
light a fire.
I'm going to be hungry. I'm just saying I'm going to feel peckish and
Well,
thirsty.
Yeah, but it's not just about you, Jim.
Well, you'll probably want a snack.
What have you got?
I've got the pistols.
Oh, pistols.
What we're going to shoot?
You.
Apparently we're going to need the pistol because if we run out of food, Julie's going to get hungry and she's going to want to shoot me.
I'm not going to want to shoot you because I might get hungry.
Just because you want to shoot me because I'm getting on your nerves. Fine. As far as we're concerned, this is all about, you know, whether we can survive on the moon until we're rescued. But it's clearly about, actually, whether we're cooperating.
We're quite good at that.
It might not seem like it all the time, but we actually do manage to compromise and cooperate.
What do you mean, doesn't seem like that?
Well, it just... Sorry, dear.
While all this is happening, our facial expressions are being recorded, and a team of evolutionary psychologists are analyzing our results. Then they call us for a meeting.
So we've been videoing you while you've been doing all of these tasks, and we've been measuring your facial movement, whether you furrow your brow, whether you raise your brow, whether you smile, whether you wrinkle your nose.
Funny look, isn't
Yeah,
it?
eyebrow raising.
Eyebrow raising.
So this gives us a huge amount of very detailed information, of behavioural information that's happening during a normal conversation.
Jim has a rate of approximately 127 muscle movements per minute.
Is that good?
That's very high.
Quite high, yeah.
The average is about 100.
Right.
Julie had slightly less facial movements than me, but it might be because she tends to play her cards close to her chest.
I might just be one of those really deadpan people.
The poker face.
This project is investigating how much we use our facial expressions to bond with others, especially our nearest and dearest, and comparing it with other primates.
Many primates have facial expressions that are similar to ours, but our faces are the most expressive. And we think this sort of complexity is probably really important to humans as a species, and how we manage these really complex, intense social interactions we have with other people.
That's helped the evolution of our brains, the fact that we are able to communicate with facial expressions.
That's the basic idea behind the social brain theory, that what is difficult is the social stuff. So forming